All right. Well, welcome everyone. I see people trickling in here. So, my name is Paul Meyer. I'm the editor of Acres USA magazine, and we're excited today to have this webinar with Dr. George Taylor from Enlightened Soil, EnSoil Algae, and he's going to be presenting on food as medicine. And I'll just do a little plug as well. Dr. Taylor has written an article that's going to be in the January issue of our magazine that will go over, I'm sure, a lot of this same topic. And I just got through editing it the other day. I think it's a great article that will be informative for lots of people. And I just want to also plug real quick our 50th annual Acres USA EcoAg Conference. It's 1 through 4 December in Madison, Wisconsin. And we'll put the link to that in the chat here. It's just conference.acresusa.com. Lastly, if you have questions, Dr. Taylor is going to present for about 30 minutes or so, and then we'll have some time for Q&A. If you have any questions, there's that Q&A block down there at the bottom of your Zoom screen. So just click on that and throw your question in there, and we will get to it at the end here. So with that, Dr. Taylor.
Okay, Paul, thanks a lot for having me. Uh, Food is medicine. Yeah, big movement. What I'm going to do today, if you're interested in this movement or working in it, I'm going to give you some ammo. I'm going to talk a little bit about the biology, the medicine biology, and uh, yeah, ammunition for your argument. Let's start with the conclusion. It's the phytochemicals, which are antioxidant and anti-inflammatory, that are responsible for medicinal, nutraceutical effects of food. And in this review, I'm going to talk about the cardiovascular medicine. Uh, why not food is medicine? Well, a lot of food isn't that medicinal. A lot of stuff that's grown conventionally isn't. Stuff that's grown with regen ag is. So I've always thought as medicine makes sense. My qualifications: I'm not a farmer, not an agronomist. Interested in soil scientists, but certainly not a soil scientist. I've had a long career as a cardiologist, taking care of patients, but the majority of my career has also involved teaching in medical schools and doing clinical research. Why cardiovascular medicine? Apart from the fact that I know something about it, it's the most common cause of death in the United States, much more so than in cancer, for example. And we're going to be talking about atherosclerotic cardiovascular disease, or ASCVD. The heart, it's the size of your fist. And on the surface of the heart, there are blood vessels, arteries that are the size of the veins on your hand. They are literally fuel lines to heart muscle. Cork one of them off, tie one of them off, and the heart muscle supplied by that artery dies over the next 3 hours. Starts dying right away, but dies over the next 3 hours. The biology of atherosclerosis, or plaque formation, it starts with the bad cholesterol, LDL cholesterol, being oxidized. And oxidized LDL is able to penetrate the arterial wall. Can you see my cursor moving, by the way? Okay. Anyway, basically, the oxidized LDL penetrates the arterial wall, where it sets off a series of inflammatory reactions that involves multiple different types of white blood cells that interact with cells that are in the arterial wall, changing them. Some of them are smooth muscle cells. Complicated slide. Hey, it's a complicated process, and it's amazing how much we know about it now. The end result is a core of cholesterol / lipid covered by a cap of fibrous tissue that separates the lipid core from the bloodstream, the lumen, the bloodstream. And here are a couple of different kinds of plaques. First, on the left-hand side, the 3-layered arterial wall, the lumen is the middle part. That is a cross-sectional view, is the bloodstream. Let's look at the far right cartoon first, the stable plaque. Stable plaque has a lipid core, but it's tiny. And there are very, very few white cells, very few inflammatory cells in it. The cap separating the lipid core is thick, thick and fibrous and very stable. This thing isn't going to fall apart very easily. On the other hand, in the center panel is a cartoon of an unstable, in the business we call them vulnerable plaques, likely to cause heart attack. The lipid core is large and it is packed with inflammatory white cells. And these white cells can produce enzymes that actually chew away and degrade the fibrous cap, weakening the fibrous cap. When that fragile cap fractures, the contents of the lipid core are exposed to the bloodstream, and that immediately causes blood clot formation. The thrombus or blood clot that forms, if big enough, totally blocks off the blood vessel. Bingo, heart attack, or in the case of arteries to the neck, stroke. So the question is, what causes plaque to be unstable versus stable? There are a number of medicines that we use that actually are anti-inflammatory and promote the formation of stable plaque rather than unstable plaque. One more gee whiz fact about this that's really interesting is if you look at the cartoons, it looks like the lumen, the blood core, the opening in the blood vessels is a lot smaller with a stable plaque, and that's true. These plaques often narrow the vessel much, much more than the plaques that cause heart attacks. And that is why, by the way, that a screening stress test is that stress tests are only positive if you have a tight blockage. With these unstable plaques, there may not be very tight blockage. The majority of heart attacks are caused by plaques that block the vessel by less than 30%. And that does not lead to a positive stress test. So stress testing is not all that accurate for ruling out the possibility of heart attack. So there are two questions that obviously come up. What causes the start of the plaque formation process? What causes oxidation of LDL? Number one. And the second question is, what causes plaque inflammation? The acute problem with plaque rupture and thrombosis. And it turns out the answer to both is inflammation anywhere in the body, which we refer to as systemic inflammation. Strep throat is a systemic inflammation. Pneumonia is a systemic inflammation. An infected toe is systemic inflammation. So basically, inflammation anywhere. I'm going to focus first on the acute illness, acute heart attack. This is the research on this has been going on for a long time. I first encountered this research in roughly 2000, and I've been following this area since, and have actually done some research on the inflammation story. There are more than 80 published studies at this time. The latest one was published in the American Journal of Cardiology last week, talking about the increase in the heart attack rate with COVID infection. So let's just run through this. Influenza, the odds ratio of having a heart attack with influenza is 3.8, roughly 4.0, 3.8 times the normal rate of heart attack in the months after having inflammation. COVID, 4 times the rate of heart attack in subsequent months. Bronchitis, COPD doubles the rate of heart attack. Pneumonia, bacterial pneumonia, big time, an 8-fold increase in heart attacks, as it is with Staphylococcus sepsis. Urinary tract infection, not as inflammatory, but a 3 times increase in the rate. Periodontal disease, the dentists now claim to prevent heart attack, and they got something. It turns out with periodontal disease, meaning real disease, meaning loose teeth, okay, doubles the risk of having a heart attack. That inflammatory illness does increase the risk of heart attack. What about non-infectious diseases? Rheumatoid arthritis, there's a doubling of the rate of heart attack. Lupus, roughly a doubling of rate of the heart. Osteoarthritis, a 30% increase in the rate of heart attack. And it's worse if you've got more inflammation. The rheumatologist looking at run-of-the-mill arthritis, osteoarthritis, have developed what they call a misery index. And if the symptoms are more severe and the person is more miserable, the there's more inflammation and the risk of heart attack is higher with a high misery index. Asthma is an inflammatory condition that doubles the risk of heart attack. The metabolic syndrome is something that people in the food as medicine movement talk about a lot. Metabolic syndrome is abdominal obesity leading to diabetes plus hypertension and a higher risk of heart attack and stroke. It doubles the risk of heart attack, and it turns out that fat cells in the belly produce inflammatory enzymes that provoke plaque inflammation. Basically, any illness that causes inflammation can raise the risk of acute heart attack. And there is a common measure of inflammation called the CRP, C-reactive protein, a very simple blood test to do. It's cheap. It can be done in the emergency room, and the result comes back in 30 to 60 minutes. Okay. Pretty much everyone who comes into the hospital with unstable vascular disease or with an acute event like heart attack or stroke, the CRP is elevated. Okay. So the next question is, if you could avoid systemic inflammation, could you lower your risk? And it turns out there are a lot of data indicate that that's the case. For example, in rheumatoid arthritis, the CRP level predicts the risk, not just the usual risk factors like smoking. And there's a stereo effect. In rheumatoid population studies looking at rheumatoid arthritis showed the high risk of heart attack, but not in the people who are taking prednisone. In asthma, inhaled steroids reduce the risk of heart attack. And in fact, the fluticasone, the inhaled steroid, lowers the CRP by 50%. Flu and COVID vaccine. When a flu epidemic goes through the community, there's an increase in the risk of heart attack, except in one group, the group that got vaccinated. And why is that? Here are the numbers with both vaccines. If you get vaccinated, your risk of catching the flu or COVID are reduced by 70%. It's not perfect. Okay, but the 30% who get the flu anyway or get COVID anyway, they're not as sick. They don't have as much inflammation. So yeah, the risk doesn't go up in the groups that's vaccinated. The final thing on this slide is the most important, though. There was a huge study published about 3 or 4 years ago now, where they looked at a common anti-inflammatory medicine used in people who had a history of heart attack or stroke. Okay, probably I think there are between 5,000 and 10,000 people in this study. Put on colchicine or a sugar pill. Colchicine is an old-fashioned drug used for gout, and it blocks the action of white blood cells. And they found that the group put on colchicine had a 31% reduction in heart attack over the next, well, the follow-up period was 3 to 5 years. Okay, let's switch gears and talk about the lifetime risk, the chronic risk, the development of atherosclerotic disease. And it turns out that inflammation early in your life increases the risk of you having a heart attack when you get old. How can that be? Well, it's because inflammation early in life, this is back to that first slide, inflammation early in life oxidizes LDL, which gets the process started, okay? And there are a number of very interesting examples of this happening. This one is really, really interesting. It turned out in the mid-20th century, the rate of heart attack doubled in the United States and really all over the world, okay? I came on the scene right about here in the 70s, okay? So when I started practicing medicine and cardiology, we were in the middle of a heart attack epidemic. We knew it, that there was a doubling of the risk, and we didn't know why. No one could explain why it went up. You know, the epidemiologists looked at changes in diet. They looked at access to ready-made cigarettes. They were never able to explain it. And then lo and behold, late in the century, it went away. And now if you take the slide out further, now it's back to normal, back to the baseline risk back here. What happened during that period of time? A lot of answers, but the one that's gaining favors the most likely is the influenza epidemic of 1918. It killed 50 million people worldwide. Compare that to COVID, which killed 7 million people worldwide. The 1918 Spanish flu was extremely inflammatory, and it targeted young adults. So typically, a young kid in boot camp would catch the flu, have a cough and fever, would be admitted to the hospital, and be dead in 2 to 3 days. That's how it worked. They went in with a cough and fever and then developed lung congestion because of intense lung inflammation, and they died from the respiratory failure in 48 hours. Now, the people who survived the flu were left with a legacy. They were left with a legacy of oxidized LDL, okay, from that inflammatory hit that started the atherosclerotic process. So that when these people reached the heart attack ages of 40, 50, 60, 70, yeah, they started having more heart attacks because they had more atherosclerosis. It went away. Why? Because those people who were 20 years old in 1918 were gone by the beginning of the 21st century. They died out. Because they died out, the heart attack epidemic disappeared. There's similar examples of this from other countries. For example, in France, in the late 1700s, there was a highly inflammatory epidemic that killed young children. In retrospect, it was probably measles, which is a big time inflammatory illness. And when that generation made it to age 70, they have the data. They made it to age 70. They died with heart attack more frequently. Interestingly, when that same generation reached their 20s and were being inducted as draftees in Napoleon's army, they were also shorter. Okay, so the bottom line is that we've got good data now showing that any inflammatory illness spills over into a vascular inflammation that can cause heart attack and stroke, both in the short term, systemic inflammation provoking plaque inflammation, heart attack within a year, and lifelong. There's a lot of talk about why we live longer than our ancestors. People talk about better diet. Wait a second, better diet? Look, that heart attack epidemic in this country went away, even though the obesity epidemic has developed. Okay, diet wasn't the answer. One answer, and people are buying this pretty much now, is our lifelong inflammatory burden is lower. We get treated for strep throat, we get treated for any infectious illness. We just don't, have better dental care. Okay. Vaccination has to play a role. So we just don't have as much inflammation. By the way, I'm talking about vascular disease. Why? Because it's so common, but it's also what I know something about. A cancer researcher could tell you exactly the same story about oxidation and the development of cancers. Okay. So how can food be medicine? Well, since the disease results from oxidation and inflammation, the question is, are there antioxidants and anti-inflammatory agents that can block the disease process? Are they in food? Is there evidence? What's in plant foods? Okay, there are three general things. Sorry for being simplistic. First of all, is calories, carbs, fats, and proteins. Then there's a group of compounds in plants, signaling compounds that regulate plant growth, like auxin, cytokinins, gibberellins. We call them signaling compounds. The botanists call them phytohormones. I tend not to use that term because the EPA doesn't like the word hormone in anything, but that is what they're called by botany. Then there's a third group of compounds in plants that are now referred to as phytochemicals. And they've also been called micronutrients, but they're probably not really nutrients. Polyphenols, flavonoids, and others. These are the organic compounds in foods that are responsible for plant color, odor, and taste. And when plants have more of them, yeah, they're a brighter color and they taste different, taste better. Just as an aside, the biostimulant EnSoil Algae increases the phytochemical content in food, and cattle, if they have a choice, will graze on pasture that's been treated with the algae rather than algae grown with chemical NPK. But let's continue with these slides. So is there an ag practice or an input that makes the food healthier? And I'm going to talk about two. One is a paired farm study, and another is the Montana cattle observation. This paper, published in 22 by David Montgomery and his colleagues, I think is going to turn out to be a landmark paper in the food as medicine movement. Okay. Here's what they did. And it was a major effort, They collected crops, plant food grown from 20 farms, 10 pairs of farms. Half of them, 10 of the farms, had been managed with regen ag practices for, the paper said, more than 5 years, 5 years or longer. The other 10 farms that were controls were managed with conventional ag, chemical NPK. Okay. They looked at these 20 farms were spread out over the United States, and they collected food from all of these farms, and they showed that the food from the regen ag farms had increased nutrient density. They also showed that they had an increase in phytochemical content. This is text from their abstract, and they said that their claim is that soil health, microbial mass, and biodiversity in soil was responsible for the increased nutrient density and for the phytochemicals in food that are not conventionally considered nutrients, but are relevant to chronic disease prevention. Okay. I think they're right. They identified soil health, i.e., micronutrient mass and diversity, as the source of these phytochemicals. Did they show a health benefit? When I've read this paper a dozen times, and I think I discovered that they did. They didn't talk about it much in their paper, and I hint at it with this last sentence, which I unfortunately cut off. But basically, they in some of these farms, they were able to get meat, beef and pork, that was raised using regenerative practices, regenerative grazing practices. And they showed that meat had a more favorable fatty acid profile. Okay, I'm claiming that measurement is basically a health benefit, a measurable, documented health benefit. Another group of evidence. Brian Massard from Remnants Angus near Dillon, Montana, big Angus cattle operation, has been using the biostimulant, the live algae biostimulant, EnSoil Algae, for a few years. And he came to us probably 8 or 10 months ago, and he said the cattle are healthier. We said, okay, what are you talking about? Yeah, is there, they look healthier? Oh, they look healthier. Do you have anything you can measure? Anything that we can document? And he says, yeah, there are some things that we measured. We found, he said, the heifers have a lower AI, artificial insemination failure rate, a lower open rate. Normally, the open, and by the way, he discussed these data 1 month ago on an acres uh, webinar that I would encourage you to listen to because, first of all, his firsthand description of the data, but also is hugely entertaining. Okay, very interesting. Anyway, his AI failure rate fell from normally 15% to 5%. In addition, he normally has to use a lot of mineral supplements uh, based on the mineral content of his forage. He noticed that the cattle were ignoring the mineral supplements, and basically his mineral supplement use has declined by 90%. And he also said that the weight of his heifers in the early spring was roughly 100 pounds heavier. Okay. Uh, Measurable data from this ag input, a live algae biostimulant. And by the way, a huge amount of soil testing all over the country with the use of the live algae biostimulant has consistently documented an increase in soil health, particularly with Haney testing, an increase in soil respiration, and with PLFA and genomic testing, an increase in microbial biomass and diversity. Okay, I'm on time, which is good. Let me with this as background, let me make some conclusions. First conclusion is, yeah, it's the phyto. I think it's the phytochemicals that are the story here. I think it's the phytochem. If food can be medicinal, it's because of a higher phytochemical antioxidant, anti-inflammatory content. Where do we stand? A lot more data. This is all brand new stuff. A lot more data are needed. Okay. What we're doing right now with the EnSoil Algae experience is we have collected crops from around the country, looking at multiple different plant types, row crops. Heck, we've got tons of strawberries and grapes and nuts and things like this. Where basically we are about to do phytochemical measurements. We've collected the samples. We're getting ready to run the analysis. So more food research is needed. uh, we talked with people at the University of Nebraska about doing a randomized study with cattle to confirm to do in a controlled fashion, confirm the cattle experience from Montana. Here's another question. Does this mean that we now have a metric for food healthiness? Here's my prediction. I think in 5 years, as this all develops, I think in 5 years, people are going to go to the store and be able to look at packaging, and they will list the polyphenol content of the food, or maybe they'll have a polyphenol or an antioxidant, anti-inflammatory index that they develop from the polyphenol content that can be measured. It's measurable. And that's what they're going to pay money for. There will be instead of just saying health food is healthy because it doesn't have X, Y, or Z, they're going to be able to say it's healthy because we've measured it. We've measured the stuff that makes food healthy. Another comment. Do soil health measures like microbial mass and diversity, if they really are responsible for the phytochemical content, it may be that we're going to be able to predict the medicinal value of a food based on soil testing. Okay, based on Haney testing, for example, or genomic or PLFA testing. All right. Here's another possibility. So biostimulants, if they can boost soil health, i.e., boost organic matter and soil respiration, microbial biomass and diversity, can they reliably boost food's nutraceutical value? Okay, health value. Uh, Brian Massard in Montana was not using regen ag practices, although, of course, he'd never tilled his native grass pastures, a lot of it. He used an application year of algae and had done that for 4 years. Uh, but it's not a traditional regen ag movement. Uh, Regen Ag seems to be able to do this in and of itself. Would Regen Ag plus the algae be better? Are the two together better than either one of them alone? I guess they would be, but we don't know. That's where the research is going to be heading, I'm sure. And finally, here's a question for growers and in the economy, the grower economy. If a grower is producing healthier food and it's measurably healthier, okay, look at number two above. It has the health metric. First of all, it won't be many of them doing it. Uh, Regen Ag is less than 5% of growers right now in this country. And use of the live algae biostimulants is just developing, okay? It's really a minority of growers who are doing any of these things. So their food is going to be their food, measurably healthier food, is going to be in short supply. So, if supply and demand means anything, the market will reward those growers. Uh, uh, I'll stop at that, and we can have discussion.
Great. Well, thanks, sir. That's really great. And like I said at the beginning, a lot of this correlates to the article that you wrote that's going to be in the January issue of our magazine. And um, yeah, it's really a fascinating topic. Um, I guess maybe one of the questions that I would ask just personally first is, what do you envision as, I think it was point two on your conclusions there about metrics that are going to be on products in the future? Do you envision this as like some kind of single index, or uh, are there going to be a number of different, things
Yeah.
that are going to be tracked on nutrition labels going forward? What do you think will happen, and what do you think the best thing would be?
How are people going to learn about this is one thing. Okay. I mean, the consumer, how will the consumer learn about this? I think grocers are going to teach them, they're going to market. Let's look at the cattle experience in Montana and look at a human corollary to that experience. Who's the consumer in that experience? Well, it's the cattle. Okay, it's the Angus heifers. Who is their grocer? Well, it's Brian Massard, who's supplying their food. Uh, Brian Brian speaks for himself, and he talked about this on his recent webinar. But he's now asking, i.e., requiring uh, local growers who supply him with hay and grain. He wants that grown with algae. Why? Because his consumers are healthier, i.e., his consumers are demanding it. The middleman, the grocer, is going to be kind of in the driver's seat. And when the grocer realizes he's got something he can market to consumers that is understandable, he's going to market that. And that's how consumers are going to learn about phytochemicals. We're going to have people talking about phytochemicals or polyphenols, flavonoids and carotenoids. Uh, Probably the easiest way to do that, of course, would be not to uh, list all of these different compounds. Uh, and oh, by the way, I came across an article last week showing that resveratrol, which is a polyphenol that's included in grapes, has been studied and shown to prevent LDL oxidation in an animal model, okay, in a mouse model. Resveratrol, a polyphenol, less LDL oxidation. Does that mean it makes the animals healthier? No, it just means that their oxidized LDL goes down. All right. That's not the same as a health benefit. But it's, there are a lot of data on these polyphenols. But basically, when health benefits have been documented, even in animals like cattle, grocers are going to catch on to this. And it's going to be an opportunity for them to market their product. They're going to want food rich in these compounds, and the growers who grow them should be able to sell them for a premium. But they're going to be teaching consumers about it. Will they list all the chemicals? It may be easier for them just to develop an industry-wide phytochemical index or an antioxidant index, something like that. But that's all going to be developed, I think, over the next. I think David Montgomery's paper and then the subsequent cattle thing point us in that direction, that people are going to learn about these compounds, and what it's not just about food as medicine, or it's about what makes food medicine. And it's going to be what makes food medicine that's going to sell the food. It's not just going to be the title food. This is medicinal. No, it'll be more sophisticated than that. It'll be what makes food medicine, and that's what this was going to sell.
Great. Yeah, and again, if you have any questions, go ahead and throw them in that Q&A box. I'll just ask one follow-up on that. Do you see a role for the FDA to mandate, like, here is the index of flavonoids or whatever that need to be on nutrition labels? Do you think that, kind of pushing that from the top could be helpful in this case?
Uh, I'm not a fan of government doing everything, but quite frankly, I never thought of that. Uh, who knows?
Yeah,
Uh, I don't know
sure.
how it'll develop, but I'd love to see it develop as a consumer movement instead. But who knows?
Yeah, yeah. Here's a question that is kind of more directed to Dr. Taylor, the cardiologist. It says, I have a keen interest in leafy greens, kale, and spinach with high lutein content. Phytochemical lutein is known for good eye health. It mitigates AMD and cataracts. Do you know of any clinical data that suggests it's another tool to mitigate heart disease?
Yeah, uh, that's a great question, and it's uh, the answer is that there is no answer. There is no demonstrated benefit. Um, there is no demonstrated benefit that the leafy greens or leucine have an eye health benefit. What there is, there are studies that have shown that leucine or other polyphenols, for example, antioxidants, have a benefit on uh, chemical processes that have to do with eye health, or have a benefit uh, on things that affect eye health, say in rat studies or mouse studies. But there are no studies, believe me, that have ever been done showing that leucine or anything else in any food affects the health of affects human eye disease. All the data that they talk about, you got to back up. It's not data from humans. A lot of polyphenol studies have been done showing it blocks oxidation of LDL. Okay, now people take that and they say, okay, it prevents heart disease. That's not the same as showing that the compounds or the polyphenols prevent heart disease. No, it doesn't. That's not the same as showing that it prevents disease. It's showing that it lowers raises lowers oxidized LDL, but that's not the same as showing that something prevents disease. It's really hard to show that something prevents disease. A lot of people in the food as medicine are very interested in the metabolic syndrome. So they talk about doing studies where they take people with the metabolic syndrome, fat and diabetic and obese, and they want them to get into diet studies, okay? Showing that if they eat that right Mediterranean diet, leafy greens, whatever, that if they eat that diet, they start doing better. You know how many people it takes to show that a clinical benefit for anything? I mean, we're talking studies of tens of thousands of patients. It routinely costs billions of dollars. The first approach to this is going to be done with animal trials. For example, we've been talking with university researchers about doing a study with a mouse, a strain of mice that develop the metabolic syndrome when they're on a high fat diet. These mice actually become obese and diabetic. Okay, and so the question is, if they eat food with a higher phytochemical content, in our case, food that's been grown with the algae, will they do better than they would than another group of the same type mouse feed with the same kind of food grown with chemicals? That's manageable. That's kind of a controllable and manageable study. You know, to shift from that to a human study, golly, listen, I've been involved in clinical research, involved in these multi-center trials for 30 years, and I will just tell you that is that's the last kind of study you do after you've done all the preliminary stuff, because it's just impossible to do. I mean, hard to do those studies. That colchicine study that I told you about, that where they gave half the people colchicine, half the people sugar pills, probably close to 10,000 people in the study. That study had to cost close to $1 billion to do, just to give people a sugar pill versus the other pill. Unbelievably hard research to do. So
Mhm.
yeah, to get back to the original question, no, leucine and kale. Hey, listen, I eat kale every day, uh, but leucine, evidence that it affects human health. There is not any evidence in humans that it has a health effect.
Good.
There's evidence in animals maybe that leucine does something, but not evidence that it changes eye health in humans.
Yeah. Good. One more quick medical question, and then I think some people wanted to ask some questions about the algae itself. Um, is there a correlation between dental plaque buildup and cardio plaque? Is that in any way related?
You know what it is? It's really the inflammation itself. Okay. The I did a study, a preliminary study where I looked at people who came into the hospital with unstable chest pain syndromes. Uh, Half of them turned out to be a false alarm. Okay, and the other half, they actually did have unstable plaque. That we could tell that. All right, and we looked at these people for a history of inflammation, and yeah, one of the common things that the people who had the real disease turned out to have was periodontal disease, meaning they had loose teeth or had lost teeth in the preceding few months. Okay, not just the fact that they had a lot of plaque, but yeah, that relationship doesn't have to do with just the plaque itself. It probably has to do with the plaque as an indicator of underlying gum disease that accompanies plaque. And if that gets really bad and causes system-wide inflammation that spills over into plaque inflammation, yeah, it increases the risk of heart disease. Let's talk about what that risk means. Does it mean that if you got periodontal disease, you're going to have a heart attack next month? No. These are population effects. Okay. Maybe the chance of you having a heart attack next month is 1/10 of 1%, or next year is 1/10 of 1%. Well, if you've got one of these inflammatory conditions, maybe it's 0.2%. The risk is still low for the individual. These are population data, but it's the population data that really do identify disease risk. So
Yeah. Good. Another question, what labs do you recommend food testing for antioxidants? But really, the question I think is what percent increase is valuable to the consumer? 10%, 25%, 50%, etc.
we don't know. All of this is brand new.
Yeah.
Okay. I mean, all of this is brand new. I I've been reviewing this scientific literature for at least a decade now, and I'll tell you, there isn't much science proving a health benefit of so-called health food. Uh, Wait a second. Well, gosh, if you take pesticides out of food, it's got to be healthier. Yeah, I agree, but there's no evidence proving that taking the pesticides out of food actually makes you healthier. Hard to do those studies. The two things that I have found are, number one, the Montgomery study, which I think is going to be extremely influential in the development of this whole area of interest, scientific interest, and then the cattle study, which are observational, observational data, but pretty darn convincing. All of that needs to be developed. So we don't know we can't talk about percentage increases or that are going to make a difference. There are a lot of labs that do phytochemical testing. It's not cheap to do. We're doing a study using a New York lab that has been doing this kind of work forever, and to do the two tests that we need to do is going to cost $500 a sample. We're going to be running probably roughly 20 samples. So this is not cheap stuff to do, and it's not the stuff your local food testing lab can do. This is all specialized organic chemical measurement stuff, mass spec, and so forth. So
Yep. Good. And then a couple questions on the algae itself. What makes the algae different or better than mycorrhizal fungi or bacterial applications or minerals?
yeah, look, this is all being researched as well, but uh, the live algae are in the biostimulant category of soil amendments and plant inputs, ag inputs. A lot of the original research that was done on biostimulants was done using live algae. They grew the algae in a tub and then it applied it to the fields. That never developed commercially because nobody could keep the algae alive. So the biostimulant industry right now is made up of a lot of derivatives or extracts, many of them extracts of green algae, the stuff that we use, all right, or seaweed. Seaweed is an algae. The seaweed that you use is basically uh, an algae preparation, but it's dead algae. It's not the live algae. The live algae go on the soil, stay alive, and continue to produce the phytohormones that we think cause their effect: auxins, cytokines, gibberellins, salicylic acids, things that have to do with plant immunity. All of these compounds are produced by the algae. They're recognized by soil microbials. We consistently measure an increase in the mass of the soil microbials and in the diversity of soil microbials. Dr. Montgomery and colleagues have identified this biodiversity as the source of the phytochemicals. Okay, we agree. It's the probable source. But what we were able to do is figure out a way to keep the live algae alive so that it can be put on live and work like the original biostimulants work. Now, another thing that's happened is if you look at most biostimulants that are commercially available, they don't have much data showing an improvement in soil health, meaning the increase in better soil respiration, increase in microbial mass or biodiversity. The live algae seem to be more effective in that part of it. So
Good.
yeah,
Yeah.
we think it works better than the dead algae does or the extracts.
Yeah. And someone else is asking, can you compare it to duckweed or seaweed you already
Yeah,
answered, but yeah.
absolutely. Yeah, no, yeah. Like seaweed is an algae. Okay, it's a macroalgae, not a microalgae, but basically it's an algae, has the same stuff in it. If you put it on in large enough quantities, it works like fertilizer. It actually provides nutrients. plant nutrients. The algae is a biostimulant, and the live algae is a biostimulant, goes on in micro quantities, okay, and produces compounds that influence the natural processes of fertility and growth. It's not itself a fertilizer. There's not enough biomass for it to be a fertilizer. It just stimulates the natural soil processes that create fertility. That's what a stimulant does.
Great. Another question, does phytochemical content in plants also correlate to plant health and yield in the crop?
Well, it's interesting in looking at this. Yeah, when the live algae is used and it's apparent that the phytochemical levels hot going up, yeah, yield tends to go up too, and it's crop dependent. Heavy feeders still need some nitrogen input, but we've shown that the the nitrogen input can be reduced, say with corn by maybe 35% by adding the algae without any change in yield, without any loss of yield. Okay. So yeah, the biostimulant does have a good effect on yield and plant health. It's interesting; it has a big effect on color and on taste. A number of ranchers have told us that if they have pasture that half of which has been treated with the algae and the other half is not, the cattle graze on the half that's been treated with algae. Brian Massard tells an interesting story about uh, hay bales that are roughly a year old and they look moldy and fermented, etc. That he didn't think were going to be any good. The cattle wouldn't care about them, and he went ahead and threw them out anyway, and put them next to hay that had been grown conventionally, not with the algae. And he said the cattle chowed down on the moldy hay bales. Why? Was it because it was healthier? Or did they graze on the algae treated grass because it's healthier? Don't think they're that smart. I think it's because it tastes better. And it's the phytochemicals that are responsible for taste.
Mm-hmm.
We're doing studies in strawberries, and the people out in the field who've been collecting these strawberries, I ask them, is there a difference in taste? And they say, yeah. And I say, is it the sweetness of the strawberries? They say, no, it's not sweetness. It's the strawberry taste. One person said, it's the strawberryness of the strawberries.
Mm-hmm. Yeah.
Interesting to think about what that might do to wine. If there's an increase in the phytochemical content of wine, we don't know. May make it better, may make it worse. We don't know.
Yeah. There are a couple questions about uh, the longevity of the algae in the soil. I think both are coming from kind of the plains, but or Texas. Will the algae stay in the soil if it only gets 10 to 12 inches of rain a year? And then
Yeah.
also, have you seen evidence of the algae after application?
Those are such great questions. And again, uh, we don't know. The Chlorella vulgaris, which is what we're using, is a pretty tough bug, and it naturally resides in soil. By the way, we're growing it in water, and it's a freshwater algae. But yeah, there's plenty of it in soil. Our people who are the phycology people, the algae specialists, the PhDs, say yeah, they think that it lives for a while in soil. One thing that may point to the fact that it lives is the fact that say for pasture, one or two applications during a growing season is enough to have an effect on yield, okay, and on growth, suggesting that it's more than just, if you put if you put down a concoction of gibberellins or auxin, the phytohormones themselves, which have been extracted, if you put that down, basically it's just like taking a short-acting medicine. It's gone before tomorrow. Okay. But if you take a long-acting pill, maybe it hangs around a while. And the fact that it seems to have this extended action in practice, you know, say on pasture or on cornfields or on bean fields, suggests the action lasts for a while. Again, a lot of research needs to be done in this field. We just don't know for sure. Uh, I guess that's the best answer I can give on duration of action right now.
Oh, that's great. I definitely appreciate you. You come over as a true researcher. Your a lot of answers are we can't say. That's yeah, that's a good honest answer. A lot of people wouldn't don't necessarily always give that answer, so I appreciate that. Uh, do you know of any movement from farmers to seek formal dialogue with USDA or FDA to perform research on this to basically mimic the Montgomery study that practices lead to higher phytochemical content in crops?
Nope. You know, this cries out for university research. The Montgomery study is observational. 10 farms that were regen and 10 farms that were conventional. That is a small n. That's a small number. And it's hard to develop reliable statistical evidence with numbers that small. That said, it took a massive amount of research for our work for them to do that study, if you think about it. Going to 20 farms all over the United States and collecting all of this and then measuring it. I'm telling you, it was a massive study, a huge amount of work that they did. Uh, yeah, it needs to be repeated. It needs to be expanded. And this is, as this story gets out, what will happen is that people in nutrition and in agriculture, the academics will basically start doing the studies. I mentioned those studies that the cattle cattle feeding institute at the University of Nebraska is talking about doing. Yeah, there are going to be people repeating this, nailing
Good.
down the results.
Yeah, good. Hopefully. And then I think we'll wrap up with this. We're right at an hour about a couple questions about composting with the algae. Um, this is from a market gardener. Besides adding the algae to compost extract just before application, would applying algae to worm compost or fully cycled hot compost be of benefit?
Yeah, I wouldn't add it to hot compost. It probably wouldn't survive it. Okay. But a lot has been done combining with the algae with other organic inputs like worm casings. Okay. There are a lot of, yeah, there are a number of growers have been doing this. Notably, Russell Hedricks from Hickory, North Carolina, has been doing studies looking at combining the stimulant with the nutrient source or with the bugs themselves. And yeah, he's saying, yeah, there's some definite benefits from doing combination therapy. And let me just, I know we're at the end. Let me just end by making one comment. Saying I don't know is something I've spent a career doing with patients.
Hmm. Yeah. Yeah, no, you have to, unfortunately, but for better for worse. Well, definitely appreciate that. And yeah, we'll just wrap up by can you give everybody where to find out more about InSoil Algae, insoilalgae.com? I
Yeah, it's the website's pretty informative. It includes
yeah,
all of the research that's been done, but it also includes descriptions from farmers about their experience. And those I think, are very useful. Uh, EnSoilAlgae.com. That's it.
great, great. And again, we're at acresusa.com or conference.acresusa.com is our upcoming 50th acre EcoAg conference, and InSoil will be there as a presenter, and they'll have a booth there, and we're really looking forward to that. So, thanks again. This is being recorded, and we will send out a link to everybody so that they can rewatch it. It'll be on YouTube and within the next couple days. And thanks again, sir. Have a great day.
Bye, everybody.
Bye.